Hello there and welcome to Bio 157. The last presentation that we looked at, of course, was dealing with the process of protein synthesis, which again, as you go ahead and take a look at protein synthesis, this is going to be a way again that we create proteins by way of going ahead and assembling amino acids. And so as you look at the process of protein synthesis, like I said, the assembly of the amino acids is absolutely critical. And if you recall, we happen to have two steps to protein synthesis. We have to first write it out, which is the process of transcription. And when you take a look at a transcript, right, a transcript happens to be kind of a written record, if you will, of your grades. So this is transcription, writing out, again, where we take the DNA and we convert it into what is known as messenger RNA. And then the last part, which happens to be translation, and in translation, we have to take what we wrote out, which is the messenger RNA, and again, using the tRNA molecule with the associated amino acid, we're going to go ahead and bring this message to, again, the ribosome, and that ribosome is going to bring in these corresponding or complementary tRNA molecules with the, again, corresponding amino acid to assemble the protein. So that, again, happens to be our process here of protein synthesis. And again, if you keep hearing me say this, I always have mentioned it in several videos now, which happens to be that a gene is nothing more than a section. So a gene is a section of DNA that is going to code for a protein. So it codes for a specific protein. Well, today what we're going to be looking at is basically how do we go ahead, and if you think about, a switch, so right here happens to be my little light switch, if you will, how do we go ahead and either turn the light switch on, or how do we go about turning the light switch off? And just like in a house, you happen to have, these little simple toggle switches, and some of you even happen to have these dimmer switches, and some of you even happen to have, like a Wi-Fi switch where you can go ahead and you can have maybe an Alexa, device go ahead and, turn your lights on for you. So there's a lot of different ways here that we can turn genes on and turn genes off. And there's a lot of different ways that end up controlling genes here through a variety of different switches. And if you were taking a graduate level biology class, you would definitely go through all of the different ways. But, we're not a graduate level biology class, so we're just going to kind of skip a couple of these, if you will, and hit the main ones that are kind of the classic ways to look at how genes are controlled. Now, the reason why we want to go ahead and control genes, same thing here, right? You don't always want to have your lights turned on. You also don't always want to have your lights turned off. And so this is all about saving, if you will, the currency or energy, if you will, within the cell. And so that's why we want to do the control, is all about saving energy, saving ATP, saving resources like amino acids. We just don't want to be burning them if we don't happen to need them. So that's the big idea here with this whole concept behind control of gene expression. Now, as we take a look at the control of gene expression, we're going to talk about the classic ones that are found within prokaryotic cells. And so when we're talking about the classic ones within the prokaryote, typically we're talking about negative and positive transcriptional control. So that's what we're going to be looking at with that. And again, when I'm talking about transcriptional control, I'm talking specifically about the first step within protein synthesis. And then I'm going to go over a little bit later on here the promoter and the operator with, again, within the DNA molecule. Sorry about that little pause there. I actually had to sneeze, and I'm sure you didn't want to hear about that. So anyways, like I said here, we're going to be looking at, the DNA and adding some different parts here to it, some parts that come before the gene and some parts that come after it. And we'll talk here about what these parts happen to be in a little bit. And again, the huge reason why we want to do gene control is all about saving materials, right? Saving the materials that come from metabolism, and then saving energy. Again, when we talk about energy, we're always talking about adenosine triphosphate, okay? I am very briefly going to hit this objective right here in FYI. Probably not even going to have a test question on it. So don't even worry about that one right there. So as we get started, a couple of things I'd like to go ahead and talk about. Number one, when we start talking about genes and we start looking especially at some prokaryotic cells, normally what happens is that there's not always going to be one gene to make one enzyme. In many cases, what happens is that you're going to have a cluster. You're going to have like, 2, maybe 3, maybe 4 genes that go into the production of a enzyme. And so we call that collection, that cluster of genes there, an operon. So if you hear me use the word operon, we're talking about a series, if you will, of genes that again are going to be used for the creation of a specific protein molecule, and typically that protein is going to be an enzyme. Okay. We also are going to have regulatory proteins that are going to be made to kind of allow us to either stop or to go, if you will. And when we're talking about stopping, we happen to have what we call a repressor, and the repressor is going to be what prevents us from taking. So again, if you remember the enzyme happens to be RNA polymerase. So this is going to be the enzyme that's going to take the DNA and it is going to bring it through transcription, so we make messenger RNA. But we're going to have these repressor proteins to help kind of serve as a stop sign that will either prevent this polymerase from going or allow it to actually go. And what is going to be kind of regulating those repressors are going to be these things that we call an activator. Okay? Now, why do we need these regulations, if you will? Here's an important thing to know and to remember, is that all of your somatic cells, so this goes back to the cell cycle, that when I say somatic cells, we're talking about the cells that make up your body. So we're talking about every single body cell except for sperm and egg, because sperm and egg, again, happen to be the gametes, and that goes through meiosis. So we're talking about every cell that makes up the human body has all of the same DNA. However, when you take a look at it, they do not all use the DNA within their cell. So for example, think about this. Your brain cells happen to have the ability to go ahead and make insulin. Well, your insulin is produced in your pancreas. Your brain cells do not produce insulin, even though your brain cells have the DNA within them to make that insulin. But they don't use it. And the reason why? It's all because of gene control. Because at the end of the day, your brain does not need to make insulin, because that's not its function. Its function is to, regulate the nervous system. So it needs all of the genes to regulate the nervous system, not to make insulin. Well, what makes insulin? It's your pancreas. So your pancreas is very important, using the DNA to go ahead and make insulin. And so with that, that's what your pancreas does. And your pancreas doesn't have to think, so it doesn't use the genes for thinking. Another thing, when you start thinking about this as well, you happen to have, your body here is going to produce hydrochloric acid within the stomach. Well, when you start thinking about hydrochloric acid, your nose does not leak hydrochloric acid, even though the genetic code for making hydrochloric acid is within all of your epithelial cells that happen to be within your nose. So why don't your nose cells make hydrochloric acid? It's because the genes here do not get turned on for making of that substance. Instead, the stomach cells, and there's some different cells there within the stomach, they're the ones that happen to go ahead and make the hydrochloric acid. So again, They don't do that. They make mucus and things like that, which happens to be a totally different gene, if you will. So your somatic cells have all the same DNA, but they don't turn it on. And the reason why they don't turn it on is their functions are all different. And they only turn on the genes that they happen to need, okay? So that's the important thing to understand here as we happen to be going forward. And again, the reason why the cells become different, like why does a brain cell turn into a brain cell, it's purely here because of that gene expression. That again comes from the gene control, that we turn the gene on or we turn the gene off, and that's what makes the cells look different. That's why a brain cell is a brain cell and a heart cell happens to be a heart cell. It's all because of genes. Okay. So this is huge, that you can control the genes, and when you can control the genes, that's going to change those proteins that you get there. Okay. So with that, taking a look at the structure of the entire chromosome, if you will. Okay. So again, I've mentioned this before, that a gene is a section of DNA that's going to code for a protein. And looking here, we happen to have, again, this term here called operon. And so this happens to be right here a collection of 3 different genes that code, in this case, for the gene for lactase. And so you can see those 3 genes right there, okay? And that's what we call an operon. Now, in front of the operon, if you remember, right, going back to the previous PowerPoints, I showed you the promoter, where again, the RNA polymerase, so this right here is RNA polymerase. And this RNA polymerase is going to read the DNA and turn it into messenger RNA. And again, this is DNA right here. And the reason why we know it's DNA is if you actually look really closely, you can see that double-stranded helix that happens to be on that molecule. So I don't know how well you can see it here on the video, but you can kind of see that double weaving there. And if you also look down back over here, you can also see that double weaving. Okay. So with that, here happens again to be the polymerase. The polymerase wants to bind here to the promoter region. So we talked about that with, again, the process of transcription. And it is going to come from the promoter and it's going to run along the gene. And here's a collection of genes until it gets to the terminator, or the end part of that gene. So there's the end. So again, here is the start. And there's the end. Well, in front of the promoter is a region called the operator. This is, if you will, kind of the starting line for the process of protein synthesis. Now, in front of the operator, there's going to typically be some molecule that's going to be bound on here. And that is going to be called the repressor. And in this case, when you look at this diagram, this repressor is not bound to this operator. But if it was bound to the operator, that would be, if you will, when you take a look at the repressor, that would be like a stop sign. That would be telling the RNA polymerase, hey, you can't go. So when it comes time to go ahead and do something like turning these genes on and turning them off, it all comes down to where is the repressor currently at. Now the repressor actually gets made further up the gene. There's typically going to be a regulator gene right here that is going to be actually making the repressor. So that way this repressor right here can be bound to that operator. So that way, again, this is not going to be here. So that's, if you will, kind of where this repressor protein comes from. It comes actually, again, from protein synthesis further along. So this is what this whole gene really looks like. So you happen to have, again, you've got a regulatory gene here that will make the repressor. The repressor binds to the operator, so that way it stops protein synthesis. And then if you remove this repressor, what's going to happen is that now protein synthesis is going to be able to be built, and you're going to be able to take the DNA and you're going to be able to eventually turn it into messenger RNA. And then through the process again of translation, you'll assemble the amino acids and actually build the protein molecules. So that's what's going to happen. Now, going through the first type of control, right? The first way that we control this is through what is known as negative gene control. So when you now take a look here at this diagram, what's happened, and this is the big key, is that there's no lactose. And again, this is milk sugar. So there's no milk sugar present. It's absent. So if you don't have milk sugar, the real question happens to be, do you need the enzyme for lactase? And so if you think about it, well, if you don't have milk sugar, right, do you need this enzyme? The answer is no, because remember, going back here to this slide that I had over here, it's all about saving energy. So if you don't have the sugar, why waste the energy and the materials to make something when you don't need it? So when you look at this negative control, you're going to have this repressor that's going to be made. Oh, I'm on this diagram. So the repressor is going to be made. The repressor is going to bind here to this operator. And so this right here, when this repressor is bound to the operator, this is your stop sign. And so with that stop sign, that is going to prevent the RNA polymerase from attaching. And because of that, it can't make this enzyme. And the reason why is you don't need this enzyme because you don't need to break that lactose down. And so again, why make an enzyme when it's not needed? But let's say that again, you're this bacteria cell, and you are an individual who happens to be, you drank a bunch of milk. And so you drank that milk, and in your gut you've got this bacteria called E. coli bacteria. Well, now if you drank a bunch of milk, this E. coli sitting in your gut is like, oh my gosh, there's a bunch of free lactose there. I want to eat that. All right, not that they can cognitively think. They can't think. They don't talk. So now the lactose is present. So if you have the milk sugar present and the bacteria find this milk sugar, they need the enzyme. So they actually need that enzyme lactase. So what they're going to do now, this is what's going to happen. This right here happens to be the repressor. It got made. The repressor technically was bound here to this operator site. And I'm going to try to, make a drawing here. And so it's bound, and right now the stop sign is present. However, notice this right here where it talks about allolactose. This happens to be a type of lactose, milk sugar, if you will. That milk sugar now binds to the repressor. And now that it binds to the repressor, what is going to happen here is this right here. This little repressor now is going to be removed. And it's going to go off. And now with that going ahead and going off, the RNA polymerase can now bind. And what it's now going to do is it's going to take the DNA and it is going to turn it here into messenger RNA. And so it's going to allow for transcription to occur, and the reason why is that you have milk sugar present and you need this lactose present. All because the lactose is here. And then eventually, translation will occur. And so with that, you're going to be able to go ahead and create, eventually, the enzymes called lactase that will allow you to break down and metabolize lactose sugar. When I say you, talking again about the E. coli bacteria in your gut. So that's what we get with, again, this lactose. And again, with this type of operon here, okay? And so looking at this, this is known as negative gene control. Because again, when you look at the whole presence here, this regulator right here, now that it's bound because there is no lactose present, you stopped the process. This is negative. And when you look at negative, negative stops. And again, no lactose, this is going to stop. Okay? So that's why we call this negative gene control. All right? Now, on the flip side, there are some bacteria that need an amino acid that's called tryptophan. And, you know, if you've ever been around Thanksgiving, they supposedly say that tryptophan is what is going to go ahead and cause you to become sleepy because you ate tryptophan, which happens to be inside of turkeys. And I can tell you right now, that's a bunch of BS. Straight up, the reason why you get tired at Thanksgiving time is because, you know, as Americans, and myself included, we sit down to a meal and it's like, oh my God, it is a smorgasbord and you eat everything. And you fill up your stomach, and because you fill up your stomach, the blood rushes to your stomach, and there's more blood now helping you with digestion than what there happens to be for, you know, your brain. And so you get tired. It's not anything at all about tryptophan within the turkey meat at all. So tryptophan is an amino acid that is needed. So with that, because you need the tryptophan, with positive gene control, what's going to happen is that if tryptophan is absent, this repressor right here is going to be not created. And so because of that, you're not going to have a repressor sitting over the top right here of this operator region. And because the tryptophan is absent, you need it, and the bacteria, I should say specifically, needs this tryptophan. And if they don't, they end up dying because they can't go through protein synthesis. And so with this, they need this tryptophan. So what happens here is that now, because this is needed, the green light is on. And so you're actually going to promote transcription, and it's going to occur. So you're going to take this series, this operon, and again, the operon is a collection of genes here that are responsible for making the tryptophan. And so you start making this molecule right here, and you start putting together all these different genes that come together to make this messenger RNA, and all of a sudden, boom, now you make that, again, molecule. Which is all good, right? And again, it's great that you have that gene to make this molecule. However, what happens if all of a sudden the E. coli or whatever bacteria it is, let's say they all of a sudden find tryptophan, should you keep turning this on? Well, the answer then would be no. Let's turn it off, because why make this when we can get it for free? You know, there's an old saying, you know, why buy the cow when you can get the milk for free? Same thing here in this example of positive transcription. Why go ahead and leave everything turned on? So now here happens to be the tryptophan because it's present. And again, you want to stop this process from happening. So the tryptophan now actually binds to the repressor, and the repressor now is going to go ahead and it is going to stop transcription. And with that, of course, you're not going to be able to go through the process there of translation. And so this is what we call positive gene control. Okay? Like I said, we see this in a lot of prokaryotic cells and in some eukaryotic cells, but this is a classical way to go ahead and control what is going to be made within the cell. And the same thing over here, this is the negative control, and again, very common within prokaryotes and some eukaryotes. So there's a couple of different ways that we turn the switch on, turn the switch off. Just like, again, when I came up with this analogy, and I have never used this analogy before, which is this right here, you know, having different signals here to turn the genes on and to turn the genes off. Positive, negative gene controls are just different switches, and they function for some differences within themselves. So that's what we have right there. Okay? Now, as I was saying, eukaryotic cells typically don't, some of them do some positive gene control, negative gene control, some do not. And so with eukaryotes, they have a lot of different ways to go ahead and control genes. They can control them at the transcriptional level. They can control a gene after it happens, and they can go ahead and they can control the gene after translation. So there's a lot of different ways here that eukaryotes can go ahead and regulate your genes and everything. And so with that, taking a look at it right here, again, there's a lot of different spots right here in this whole area here where, especially in humans, we can control and we can turn these switches on and we can turn them off. So it's not just, again, positive and negative gene control. There's a lot of different ways that we can do it. We can actually control the chromatin and, again, regulate that, if you will. We can also go ahead and we can interfere with the messenger RNA molecules and things of that nature. So a lot of different things there. Okay? And I'm not going to ask you a test question. And the reason why is that there are so many different options out there that it becomes daunting to go through all of the different ways that, you know, it can be controlled here. So for that, I am not going to touch that one. But again, if you were to take even a pure genetics class at a graduate, not a graduate, but an undergraduate level, you would talk about a lot of these different controls. But I'm just not going to get into it just because, again, there's just so many different ways. I could literally spend 2 lectures on all the different regulations here, and just at some point it's just not worth the time, if you will. So anyway, so those are some big areas there. Okay? So like I said, back to it, your body happens to have, and this is a big key that people, they just don't get. Your body cells, your somatic cells here, they all have the same DNA. And again, so when you think about it, you know, it's kind of weird to go ahead and think about this one, but males, you happen to have the genetic code for making some of the breast milk and things like that. Well, why is it that males don't lactate? It's because we don't turn the genes on and off for that. We just keep it in an off position. And so again, you know, thinking about this whole thing here, because every cell has, you know, this switch right here, your cells either turn them on or they turn them off. And again, your heart cells turn on the DNA for making the heartbeat. Your, again, eyes turn off the genes for making the heartbeat because they don't need to beat. So this is huge when you start looking at it. Most people think that, hey, every cell has to be genetically different, but they're not. They're actually genetically identical, which is huge when you start looking at it that way. So with that, this just, again, happens to be positive and negative gene control that I went over. And again, if you have any questions, feel free to reach out. Hope you're having a good day, and I'll talk to you later. Bye-bye.